Hello, everyone. Thank you for being here. We are honored to have Dr. Satheesh Babu. He is a professor in the Department of Chemistry, Amrita Vishwa Vidyapeetham University from the Coimbatore campus. He leads the Biosensor Research Group and Amrita Biomedical Engineering Center at this campus, and he also heads the Amrita Biomedical Engineering Center. He also serves as the research head for the School of Physical Sciences at the campus. He has his master's in chemistry from Gandhigram Rural University and his PhD from Amrita University. He has been awarded the prestigious Innovative Young Biotechnologist Award in 2013 from the Government of India, and his research mainly is focused on different types of biosensors, immunosensors, lab-on-a-chips, and point-of-care tools. We are really privileged to have him today. Dr. Babu, the floor is yours. Thank you.
Thank you. Thank you for such nice introduction. And at the outset, I thank Professor Patricia for inviting me for this lecture series. Thank you so much. And shall I share the screen?
Yes, please.
Hope you are able to see my screen now.
It's still sharing. Yes.
Yeah. So what you see on the screen is the Coimbatore campus, the aerial view of the Coimbatore campus. And as you see here, it's a clean green campus at the foothills of the Western Ghats. And the Coimbatore campus received multiple times the Clean Campus Award, what we call the Swachhata Award from the Government of India. And the Coimbatore campus is actually on the southern side of India, the state Tamil Nadu, and the city, the Coimbatore, as you mentioned. Yeah. So this, what you see here is the main administrative block of the university headquarters, which is in Coimbatore. Yeah. So before we start, I just start with a famous quote of our Chancellor, Mata Amritanandamayi Devi, known as Amma. And she is actually a well-renowned humanitarian leader. So as Amma says, life and living are not the same. A job. These alone fail to make life complete. For that, we need compassion, tenderness, a heart that knows and others. Yeah. A bit more details of the Amrita Vishwa Vidyapeetham. It is having about 300 programs and 30,000 plus students, 9 campuses across India, as you see at various parts of India, but largely concentrated in southern part of India. And we have more than 2,000 plus faculty members and 19 constituent schools. And we ranked 7th, the top university in India, and multiple accolades like the #1 in India in Times ranking and the QS ranking and multiple ranking. We are at the top. And the recent impact ranking, we ranked as the top 1 in India and among the top 50 universities in the world in various United Nations sustainable goals. Yeah. So hope you are able to see my screen, and I'm right now on the slide of point of care testing devices. Patricia, can you confirm?
Yes, we can see your screen.
Yeah, thank you. So the point of care testing devices, as we know, the testing is done where the care is needed, whether it is the outpatient clinic of the doctor or on the bedside or at home or at the a rural village where we conduct the health camp or wherever it is. And there are multiple examples of point of care testing devices, starting from the well-known glucometer to the pregnancy test strip, and the recent pandemic taught us multiple various other options like SARS-CoV-2 antigen testing and IgG based testing, and lot many things we learned in the recent past also as point of care testing devices. And if we look at the market potential of point of care testing devices, we know which is growing at a very faster rate. And by 2030, it touches something like 73 billion US dollar. And if we see the entire global market of point of care testing devices, more than 50% is held by the blood glucose monitoring devices. And the next will be the pregnancy and fertility testing kits. And there are others like infectious disease testing and hematology testing and cardiac metabolic disease testing, etc. They are at the smaller side of the entire market. And as WHO said, a point of care testing device must be assured. What WHO means is it must be affordable, sensitive, specific, user-friendly, rapid and robust, equipment-free, and deliverable to end users. Because as we know, when we develop a point of care testing device, it must be we must be able to deliver it at an affordable cost such that it reaches to the society. And obviously, it must be sensitive for the required concentration range of the particular analyte of interest. And it must be specific to the target molecule or target analyte what we look for, and it must be user friendly, because largely the point of care testing devices are used to buy primary at a primary health center where not very trained technician using the technology, or it might be used by the patient himself or herself. So it must be definitely user friendly, and definitely we need to get the result rapidly. At least within an hour, we must be able to get the result. And it must be equipment free. There should not be sophisticated equipment that required for the testing of the required for the point of care testing. Then we may not be able to implement at the site where really the care is required. And we must be able to deliver it to the end user at a lower cost and whatever the above set points. And in this talk, I will be briefly touching upon some of the devices we have developed in our center, the Biomedical Engineering Center in Coimbatore Campus. And at first I briefly touch on the biosensors. Then we tried integrating as a single device, as lab-on-a-chip type of devices, and bit on electrochemical immunoassay and how we translated that electrochemical immunoassay for a point of care application, and a bit on the bacterial detection as a point of care device, and finally on the paper analytical device. So when we say the electrochemical sensors, this is some image immediately come to our mind. We used to have a laboratory potentiostat, then that is integrated with a computer, and we normally have a 3 electrode assembly like this. So, and this is how normally we conduct electrochemical experiments. And we also started in a similar line maybe 20 years before, but slowly we started translating those experiments which we conducted in the beaker and with 3 electrode assembly, as you see here. And this example, it's kind of a frugal innovation where we developed catalytic material that specifically oxidize the glucose, and we just coated it on a small plastic strip, which is very similar in size of the commercial glucose sensor strip. And this is actually a very handmade one. And then I tried integrating that using a small IC pin. I just removed the inner plastic. Then I was able to connect that with the electrochemical station and tested with various concentration of the sample. Then also I was able to test with the uh, real samples. So these three, C, D, and E, they are representing a hypoglycemic patient sample, hyperglycemic patient sample, and also the normoglycemic patient sample. So we were able to get quantitative results with this. Then slowly we translated as the same thing using the printed electrodes. We have an automated printing system, and in which we could print the electrodes as everyone does, like a silver layer printed to enhance the conductivity. On top of it, we printed the carbon, and the silver chloride reference electrode we printed. Then the catalytic material also printed on the working electrode. Then the unindigenously developed glucometer, we were able to connect with this, and we could read the sensing. This is an example of enzyme-free glucose sensing, where we use direct electrochemical oxidation of glucose on the modified electrode. And this technology, we have 3 patents, which is one, 2 US patents and 1 Indian patent. And this technology is undergoing clinical trials, and an industry is taking this to the market right now. And in the similar line, we have also developed sensors for other metabolites like cholesterol, uric acid, bilirubin, creatinine. And so we wanted to integrate all of them as a point of care testing device. For that, we developed microfluidic system like this, as you see on the left hand side. Then we integrated that with the printed electrode, which are modified with the catalytic material. Then also we have developed a meter, a readout system, where we can see the readings of all these 5 analytes which we are trying to test. So here our intention was, just with a drop of blood, a finger prick blood, we were able to detect 5 metabolites as a point of care testing device. And you might wonder why we should have gone for a system where the microfluidics and a lot of complexities we brought in, because we wanted to have the maximum accuracy. Because as we test in the real clinical lab, where we use the solutions to test, so we wanted to bring those things, like we have uh, a microfluidic system wherein we have the provision for the reagents to integrate and also the sample. So as and when the reagent and sample mixes through the microfluidic channels, and here we place the sensors below these sensing wells. These are microfluidic wells under which we place the sensor electrode. So we get a very high accurate results from these kind of measurements. Rather, we use a dry test strip. That's the purpose we have gone for a microfluidic based lab on a chip device for these simple metabolites. And as we know, this, when we use the lab on a chip type of system, it is a classic example of multidisciplinary research, where microfluidics involve, microelectronics involve, embedded systems and programming, and also the biosensing, wherein we have the chemistry, biology all involved. So here we started the microfluidics with the COMSOL simulations, and then we developed the microfluidic systems, where kind of a meander type of channels, wherein with alternately varying diameter. And we fabricated the optimized design by using photolithography, and followed by soft lithography for the stamp to get it. And briefly, this is how we have done. At first, we prepared the substrate on which we spin coated the uh, material. Then we did a soft baking, and followed by a hard bake, then followed by exposure, and then post exposure to hard bake, and we developed using suitable solvent. So these were the multiple steps involved in the photolithography process. And once we have done that, we normally get the negative. And on top of the negative, we poured the PDMS, baked and then peeled off, such that we could develop a microfluidic system like this. Why I explain little more detail, because if there is masters and the undergrad students, they must be able to understand what I'm trying to say. So for the soft lithography, we, on top of the negative what we fabricated by photolithography process, we poured the uh, PDMS, then baked and then peeled off. Then it is stuck on a plastic substrate such that we were able to make a simple microfluidic systems, and then we connected it using a syringe so that by applying negative pressure, we were able to make the fluid to flow. And this is how it looked like. So we have a provision for the sample inlet. Then we have the reagents, because when we integrate multiple sensors, all are enzyme-free sensors, all of them may not work under same pH as in the blood sample. So we wanted to introduce the reagent with suitable pH and ionic concentration, such that each of these analyte were be able to detect at on an electrode where no enzyme or no other biomolecules are integrated. So these are the passive micromixers with an alternative varying diameter of 1:2 diameter we optimized. Then these are the detection well. Under this detection well, we kept the sensor electrodes. So just to understand the mixing, we used the colored solutions at first. Then we made the fluid to flow. And we could see that when it reaches the sensor well, a complete mixing of the A and B, both the colored solutions were possible. Then to miniature the same microfluidic system, we introduced meanders inside the microfluidic channel, so that we could get complete mixing within less than 2 mm distance. So like this, compared to the previous one where lot many turns, lot many meanders were involved, whereas when we introduce the ridges inside the microfluidics, we could see that the mixing happened within 3 turns of the meanders and complete mixing. And this complete mixing we ensured by using electrochemical studies. In this example, what we have done is we used a glucose sensor where we mixed the reagent and glucose solution outside, not within the microfluidic channel, in a beaker, and then we tested on the electrode, and also within the microfluidics, and then we compared. You can see that this black and the pink line, they are actually one with manual mixing and other on the microfluidic system, where we could get exactly same results. This tells us that within 3 turns of the meanders, we could get the complete mixing of the reagent and the sample. And regarding the readout, we used analog front end like LMP 91000 as the potentiostat. This is an example where we connected 3 sensors, and the example which I started with was 5 analog front end. That means 5 potentiostat work parallelly, and the communication will go to the microcontroller. This microcontroller is programmed for the concentration. The calibration plot is actually programmed on the microcontroller, and we get we see the reading on the display, and also this is able to communicate with the mobile device. So this is how the first prototype was looking like this. So where we could integrate 5 sensors, and these were the sample inlets and the reagent inlets, and we could get the reading with this system. And this device was able to detect the physiologically significant range of concentration for glucose, creatinine, bilirubin, uric acid, and cholesterol. And this is a small handheld device, like very similar to that of the glucometer. And this one Indian and one United States patent we got in addition to the multiple publications in this field. And so that was actually a device, a lab-on-a-chip device, wherein much of complications involved. and pretty straightforward system where we have 5 sensors integrated with the microfluidics. But the when we see the electrochemical immunoassay, typically what we use for the detection of proteins in the system, proteins like cancer biomarkers and other proteins, where multiple steps are involved in the electrochemical assay. And this schematic explains the various steps in a simple sandwich type of electrochemical immunoassay, wherein we have multiple steps are involved. For example, this schematic shows the gold disc electrode, and which is modified with the mercaptone decanoic acid, because we want to bring the carboxylic acid and just wanted to have a self-assembly of the system. And with EDC NHS chemistry, we were able to immobilize the antibody on this. Then the surface is blocked with the BSA. Then when we introduce the sample, the protein binds with the antibody. Then then we use the label. This is a pictorial representation of antibody immobilized on gold nanoparticle modified graphene oxide surface. So when we see the system, suppose even if I take this is the prefabricated electrode which I can introduce in the microfluidic system, but still then I have to introduce a sample, then I have to have a washing buffer to wash it, then I need to introduce the label secondary antibody, again I need to have another washing step, then another testing buffer. So this is how we do it in the lab. So if we want to take this as a POC device, and the user need to introduce multiple samples, and as we know, these kind of assays normally done by a very trained technician or a very senior PhD student who is able to do the assay. So when we take this as a system where for a point of care application, there is obviously a lot of chance for the errors, because lot many reagents need to be introduced by the user. Right. So what we tried here is we attempted automating this immunoassay system by using microvalves. See, we used a concept, a very simple concept like this. We introduced the flow blockers. For example, suppose if I keep the sensor electrode, a prefabricated sensor, I mean at this stage, where I have the antibody on the electrode, and this I keep it on the in the sensor well. Then suppose this is the place where I introduce the sample, and the solenoid, in this case we use solenoid valve. That solenoid valve actuate so that this channel alone opens, so that the fluid flow is possible through this direction. Once you introduce a sample and incubate the required time, then the second inlet will open, the washing buffer that flows through this, and the washing is possible. Once this is done, then the inlet where I have the label secondary antibody that can flow through the system, because this valve will actuate at that point of time, and I can make the label secondary antibody which comes on the electrode. Once it is incubated for the required time, I can make the testing solution, again washing with the washing buffer, followed by the test solution which can come to the electrode surface. Here we have used the hydrogen peroxide, because that label which has got a pseudoperoxidase activity, so we used hydrogen peroxide solution as the testing one, so this can flow. So all these multiple steps we could automate by using this kind of an assembly, wherein we have the top layer with the microfluidics with the flow blockers here, and the middle layer consisting of the another PDMS layer which is flexible, in which we actually glued the mild steel plates, small mild steel particles. Then below which I have the solenoid valve. So when the solenoid valve actuate, what happens is this one, this stainless steel that comes down, the MS which comes down, that creates the channel to open. So this makes the fluid flow to happen, as I said, with all the four. So by using a microcontroller programming, I can control the entire fluid flow based on the optimized time what we have done in the lab. So the user just need to introduce the sample and wait for the result to happen. Maybe something like a 30 minutes incubation followed by washing and another 30 minutes. Within an hour, the system will give you the result. In the sense that complete of the electrochemical immunoassay as we do it in the lab. And we might wonder why you should go for an electrochemical immunoassay, because there are a lot of lateral flow immunoassay test strips are available. Because we know when we follow this kind of a study, which is exactly mimicking as what we do it in the lab, the accuracy of the measurement will be very high, and also we get the entire electrochemical assay as we do it in the lab on a microfluidic platform. And the top layer, the microfluidics like this, and also the middle layer wherein we have an acrylic sheet which has got hole, on which we just put a sheet of thin layer of PDMS in which we glued the metal particle, magnetic metal particle. So we could get the middle layer as this one and the top layer this one. So this we place on the solenoid valve, so that we could see as and when each of these valve operate, we could see the fluid flow to happen. Say for example, in this diagram, the B diagram, we have the B valve which is open, so that we see only the when this solenoid, the solenoid corresponding to this, when this actuate, only the blue solution which flow. Likewise, when the red one, when it actuate, sorry, this one, you see the only red solution which is flowing. Likewise, you can control each of these valve and the fluid to flow to happen. So we essentially tried translating a typical electrochemical immunoassay which happens in the lab as a POC system, and where we could test with the actual clinical samples here. And this also we tried as another valve mechanism. We used a piezoelectric valve also instead of the solenoid type of valve here. Yeah. So the third part of the discussion is on the bacterial detection, because we know normally the bacteria or the infections are detected for the specific organism by culture studies, and that on an average takes 48 hours or even more. So our thought process is how to do as a point of care, how to translate it as a point of care testing system. So we have developed a couple of fluorescence based system, but still we found that the electrochemical detection of bacteria may be still simpler one, because as we know the detection system what needed for the electrochemical assays are similar compared to the optical setup which is required for the fluorescence measurement. So here in this example, what we have done was we developed carbon content dot, and which is functionalized with the antibiotic. So for example, colistin, which is specific for the gram-negative organism, that was used to functionalize the carbon content dots. Here, typically we use nitrogen sulfur co-doped carbon dot, and that was immobilized on the electrode by using agarose. Agarose gel we used to immobilize the carbon dot on the electrode surface. So we have the working electrode of the disposable strip, which is modified with the carbon content. And then we dip that electrode in the solution that contains bacteria, and we applied a pre-concentration potential 0.5 volt plus 0.5 volt for 3 minutes time. Then we run the impedance spectrum. So by looking at the impedance value, we were able to quantify the amount of bacteria which is present in the solution. And you can see once we modify the electrode surface with the cholesterol carbon dot, we could see the fluorescence of the carbon dot. That it's a visual proof for the carbon dot on the electrode surface. And these SEM images showing the various stages and how they interact with the bacterial solution. So for example, this is the bare electrode where no modification done. And when that is introduced into the solution which contains bacteria, we find that there is no bacterial cells on the electrode surface. And this is the electrode with only agarose, no carbon dots and the antibiotic. And there are a few cells on the electrode surface. But the electrode with cholesterol carbon dot on the surface, we find that huge number of bacteria on the surface. So as and when the bacterial load increase on the surface, we find an increase in the impedance. So that was actually used for the quantification here. For example, the same thing what we have seen in the previous slide as an image, we could see the impedance spectrum for the same thing here. So we find that these were the bare electrode with the E. coli sample, and agarose modified electrode and corresponding electrode which is dipped, and then we measured the impedance here. Then we have the the electrode with carbon content dot, and that is actually studied for the bacterial sample, then where we could find a huge increase in the impedance. And also we tested that particular electrode for various concentration of bacterial sample. And we found that this sensor electrode was able to detect entire range of concentration starting from 100 CFU to 10 to the power 8 CFU per ml. So this is the 10 to the power 2 or means 100,000. These are the clinically significant bacterial load. So this sensor system was able to detect even such lower concentrations of bacteria without any culturing of the sample. What we have done is just took the sample, just introduced the electrode, and then tried measuring the impedance. And this is also tested for the other organisms. And you can see on the left hand side here, all other organisms like gram positive, they were actually giving same impedance value, but with gram negative, we find a huge increase in the impedance. And the same has been studied with polymicrobial culture also. There we found that even in polymicrobial culture, when we increase the concentration of E. coli, we find an increase in the impedance. So this clearly shows that this developed system was able to detect bacteria even in a polymicrobial sample. Then the last part of my talk is on the paper-based devices. Here, one example which I'm going to talk is on the liver function test. So here we tried testing the 5 important biomarkers in the liver function test: the bilirubin, alkaline phosphatase, aspartate aminotransferase, and alanine aminotransferase, and the total serum protein. All these 5 we tried detecting on a small stamp-sized paper strip. Normally, as we know, this is done by spectroscopic analysis or using the autoanalysis in a sophisticated clinical lab. And what we have followed was we took a Wadman chromatography paper patterned using wax printing. Then we introduced the reagent corresponding to that particular analyte. Then we used the plasma separation membrane. And also we supported with a polymer support because it's just a Wadman chromatography paper. Then we introduced the sample, and with change in concentration or increase in concentration, we find an increase in color intensity. Yeah, as samples, you can see here the bilirubin we tested from very lower concentration to very high concentration here. And we find an increase in color like this. Likewise, we tested for aspartate aminotransferase and alanine aminotransferase, alkaline phosphatase, and total protein. So these entire studies we conducted on the paper strip, and a color chart can be given to the end user for just to have it as we do with the pH paper. So this largely look like a semi-quantitative system as in the case of a pH measurement. Okay, we have made a method to convert this for the quantitative also that I'll explain little later. Once we have developed all these five analytes separately, we tried integrating them like a simple stamp sized paper strip, wherein I have 5 wells, introduced all the reagents for that. Then a plasma separation membrane was placed. Then this is actually a polymer backing to support, to give mechanical support for that. Then once we introduce the sample, we see the color change, right? Then we have tested all possible ways. We typically looked at two patterns. One is a cholestatic pattern wherein alkaline phosphatase and bilirubin concentration changes. Most of the other things will not change much. So with the help of the clinicians, we were able to fix the range of concentration what is needed, and we have tested successfully for these two. Alkaline phosphatase and the bilirubin we tested, keeping all other analytes concentration constant. Likewise, we tested another pattern which is on the hepatic pattern where AST and ALT concentration changes, and the others may not change much at that condition. So we focused our study only on these particular analytes. And also we tried testing by varying all the 5 analytes at a time. We changed all of them, the concentration of all the 5 simultaneously, and also we tested. And this paper device was able to show a quantitative color change. And we tested with the direct blood sample, as I explained. We kept a plasma separation membrane behind, and then we tested with the direct blood sample. We used a finger prick approach, and then we directly placed the blood sample. And okay, so I think one slide I missed here. Then what we have done here is we used an optical system, an optical device where we used an optical sensor, and then we quantified each of these color developed on each of these system, and that is where we could see the reading on the device, and also that is able to communicate to the mobile device. So this is a system wherein I can, instead of the $10 to $15 what we pay for the liver function test, that can be done something like $0.5 or even lesser than that. Because here the consumable is just a paper strip wherein the patient or the caregiver just need to introduce the finger prick blood sample, and using the handheld meter, we were able to quantify the color developed on the system. And this is in fact now an industry has almost done the clinical trials and taking it to the market right now. And the other ongoing similar work, I thought I will take much time. That is why I put the other thing just as a backup. This microdisc is another ongoing project in the lab, wherein we could test for the fungal keratitis, which is a very common problem in some part of our country. So where we were able to test the samples from the eye, and then we were able to detect the fungal keratitis just by using a plastic strip. As I explained, the lab-on-a-chip platform what we developed for the other one, here we could directly test the fungal keratitis, and we have thoroughly tested that with the clinical samples with the help of the eye hospital, Arvind Eye Hospital here in Coimbatore. And the other one, the second one, what I meant is we did an on-chip electrochemical lysing of the bacteria, and then we used that bacterial sample for the on-chip analysis, the molecular analysis of the bacteria and identifying what the particular organism is. And the third work which we focus on the direct electrochemical sensing and other application by using single atom based electrocatalytic material. And the last one, the lateral flow immunoassay strips, we tried develop for the osteocalcin, CTX, and other bone turnover biomarkers, where we could help the typically the menopause and associated complications. We just tried done with multiple ways like hormones, bone turnover biomarkers, and related lateral flow test strips. With this, I conclude. I I have used the work from these PhD scholars who have graduated from the lab on the left-hand side, and this is the current team. And I acknowledge the funding from the Department of Biotechnology. Most of the projects were funded by Department of Biotechnology and Indian Council of Medical Research, Department of Science and Technology, and the Mission Reach program, Mission 2020 program by the Government of India, and the BIRAC, the Department of Biotechnology. Thank you so much. Thank you. I am happy to answer any queries, concerns, questions if I know.
Thank you. It was a really great talk. Any questions? Yes, James, please.
Hi, yeah, thank you. Wonderful talk. I really enjoyed the overview of your laboratory work. I was wondering so thinking about the future, do you see other adjacent technologies playing into microfluidics? For example, I'm thinking about MEMS and other kind of photolithography driven type technologies, and whether they have any benefit for the projects you're trying to solve.
Actually, whatever the microfluidic devices which I showed you, we have done with photolithography. The Heidelberg system we used to develop all this. But recently we think that that is still costly thing, and we started working with the SLA type of 3D printers. And some of the kind of the form labs for B and all, they are able to give reasonable precision for the microfluidics application. We recently started using that, because again the problem with the cost involved in the prototyping with the lithography. So because when we started working on lithography maybe 10-15 years, where there is not much of 3D printers of this accuracy available. But now the 3D printers, we are able to do the microfluidics with reasonable accuracy. And the students get much access to the system again then.
Yeah, great. Everyone loves a good 3D printer. Fantastic.
That was also kind of my question, that have you decided to go for low cost point of care, because most of the point of care are for screening. So going for lower cost technologies would actually help. I think
So if you look at the progression of my talk, you will understand that I started with electrodes, microfluidics. Finally, I
paper-based.
concluded with the paper.
Yes.
And that too, a simple stamp-sized paper strip wherein I can use a device, so that I do the testing. 20 minutes I wait for the drying, then put it in the device so that the device will be able to quantify it. So the cost of the LFT is something like IN Indian rupee 20. That's what we visualize. It's something like $0.2.
That's impressive. Sharon, I think you had your hand raised. Do you still have a question?
Yes. No, a great talk. Enjoyed it.
Thank you.
How have you given any thought to maybe trying to duplicate the kind of philosophy and work at other centers throughout? Because in different regions, they're often plagued by different kind of problems. And I see that as a possible easy extension. So would you like to share your thoughts on that?
if I understand your questions, whether we, on a region-specific manner, whether we tried applying these concepts. That's what, correct, no, sir?
Yeah, duplicating, setting up similar such centers.
Yeah.
Yeah.
One example I can tell you, that last example which I said on the fungal keratitis, which is a common problem at some parts of the country. And the Arvind network of the Arvind Eye Hospital, they do a lot of village and rural health camps. And they said this is a severe problem and they could develop a laboratory assay for the same thing. And they wanted us to translate as a POC device. We could successfully translate it as a POC device, and wherein they just need to take the sample and put it in the device, and that will be able to, like as a fluorescence reader, it is able to, the entire processing, it is done in the microfluidic chip itself, and then finally we were able to show the fluorescence. So just rather taking so much of reagents and finally taking it to the lab for the spectroscopic analysis, we could do that. So as you said, we started attempting on it. And other one is on the recently we proposed a project which is kind of the large number of biomarkers involved in the menopause and associated complications. Like it can be hormones, it is metabolites, it's the bone turnover and other biomarkers, and lot many spectrum of biomarkers involved. So we actually proposed multiple paper-based systems for this, wherein no trained technician is needed, and the mom by they themselves were able to do, because the problem in India or in the where the rural, because in India if we see 80% of the Indian population reside in rural part of the country, where there is only maybe 20% of the healthcare is given, whereas 80% of the healthcare is directed towards the city where there is only 20% of the population. So this kind of an inverse relation we could see, and most of the rural health centers or the primary health centers, they are equipped, but largely their focus is on the maternal and contraception related problems. So nowhere these the menopause or related problems are associated, but it is something which has to be addressed. So this is what we thought, that we have to seriously do something for this, and wherein we must be able to develop technology without much of sophistication so that it penetrates to the society. So there we proposed multiple lateral flow assay systems and vertical flow type of paper-based assay systems with the readers for that. So that kind of approach nowadays we seriously look into, sir. Wherever extreme simple system that can be taken to the society, where without much of sophistication, and I'm sure we may not be able to end up with any recent publication also on this. We don't worry much about it.
Perfect. Any other questions? So thank you again. I know you're traveling, but thank you for putting this time and giving this exciting talk, and looking forward for more collaboration with you and more talks. Thank you.
Thank you so much. Thank you so much.
Take care. Bye.
Thank you. Bye-bye.